At the base of the brain, where the optic nerves meet and cross in an X of pale tissue, there is a structure so small that it would be easy to miss entirely. It rests just above that crossing point, barely touching the upper surface of the chiasm, embedded in the soft gray matter of the hypothalamus, like two tiny seeds pressed into warm earth. Each half is no larger than a grain of rice. Together, they weigh almost nothing. And yet from this place, from these two quiet clusters of cells in the deep interior of the skull, the timing of the entire body flows outward like a tide. The structure has a name drawn from the anatomy around it. Resting above the chiasm where the optic nerves cross, it is called the suprachiasmatic nucleus. The word is long and formal and carries the weight of the geography it describes, but the thing itself is small, breathtakingly, almost implausibly small. Two paired masses of roughly 10,000 neurons each, occupying a volume of around 3/10 of a cubic millimeter, which is to say, a space narrower than the tip of a sharpened pencil and shorter than the width of a single letter on this page. Nothing about its size prepares the mind for what it does. To understand the suprachiasmatic nucleus, it helps to arrive there slowly, to move inward through the brain as one might move through a building at night, passing from room to darkened room, descending toward a light burning deep at the center. The cerebral cortex is first, broad and folded and vast, the place where language lives, and memory, and the long looping thoughts of a waking mind. Beneath it, the structures grow older in their architecture and more primitive in their purpose. The thalamus, the amygdala, the hypothalamus, a cluster of ancient regions that govern hunger and temperature and hormone release, and the basic maintenance of a body alive in the world. And there, at the floor of the hypothalamus, just where it meets the optic chiasm and the roof of the mouth grows close, and the outside world is almost near enough to touch through the thin wall of bone. There, the suprachiasmatic nucleus rests, luminous and still, precise as a mechanism built to last. It does not look like a clock. Clocks have hands, or numerals, or at the very least, a mechanism that can be watched turning. What the suprachiasmatic nucleus has is quieter than all of that. It has rhythm. The neurons inside it fire in patterns that repeat with a period of close to 24 hours, accelerating through the hours of the subjective day, reaching their peak activity somewhere in the middle of what the body takes to be afternoon, then slowing, quieting, pulling back through the evening hours until deep in the subjective night they fall nearly silent. Then the cycle begins again, firing rates somewhere between 5 and 10 pulses per second at the height of the day, dropping close to zero in the deep of the biological night, and then rising again with the same patient inevitability with which the sun rises outside the skull that houses it. The brain makes rhythms constantly. Breathing makes a rhythm. The heart makes a rhythm. Electrical oscillations move across the cortex in waves that shift with attention and with sleep. But these rhythms are driven by continuous signals, by the lungs filling, by the pacemaker cells of the heart, by the chemistry of waking and sleeping. Take away the signal, and the rhythm stops. The suprachiasmatic nucleus is different. Its rhythm does not require a signal to keep it running. The clock runs from inside. This was shown in a way that still holds something of its original strangeness. Researchers isolated individual neurons from the suprachiasmatic nucleus and placed them in glass dishes, surrounded by nutrient fluid, completely cut off from every external signal, no light reaching them, no hormones from the surrounding brain, no input from neighboring cells, no indication of whether it was day or night outside the laboratory window, and the neurons continued to oscillate, Alone in their dishes, each one kept a rhythm of close to 24 hours, kept it for days, kept it for weeks. The rhythm drifted slightly from one neuron to the next, as individual cells have slightly different natural periods, but each one kept its own time with a fidelity that no other cell in the body has been shown to match. A single neuron, smaller than a dust particle, no larger than the period at the end of this sentence, ticking away in a glass dish in an artificially lit laboratory, keeping time entirely by itself, with no external world to reference and no company to synchronize against. This quality, the ability to generate a self-sustaining rhythm without any external timing cue, is what makes the suprachiasmatic nucleus a true clock rather than a mere responder to the environment. It is not a meter registering sunlight and converting it into a behavioral schedule. It is an autonomous timekeeping mechanism that sunlight can adjust but does not have to drive. The rhythm comes from within. The environment reaches in to set it, to correct the slight imprecision that any self-sustaining oscillator accumulates over time, but the engine itself runs on its own power. To understand how the external world reaches in to correct the clock, it helps to follow the path that light takes once it has entered the eye. Most of what the visual system does with light is complex and rich and slow, assembling the raw signal from the retina into the textured, colored, spatially coherent world that the waking mind inhabits, parsing edges and faces, and motion and depth across dozens of specialized cortical regions. This processing takes time and involves enormous amounts of neural tissue, but there is another pathway, far simpler and far older, that bypasses all of it entirely. Deep in the retina, mixed among the rods and cones that handle ordinary vision, there are cells of a different kind. They are not plentiful. They represent a tiny fraction of the total ganglion cell population, and they do not contribute to the formation of images at all. What they do instead is respond to light with a long, slow, sustained depolarization, a signal not suited to tracking the swift movements of visual objects, but perfectly suited to encoding one simple fact: how much light is present right now. They contain their own photopigment, a light-sensitive protein that responds most strongly to the blue end of the spectrum, to the clear saturated azure of open sky on a cloudless morning. And from these cells there runs a dedicated projection, a slender cable of axons following a route that carries no images and serves no visual purpose, running directly to the suprachiasmatic nucleus at the base of the brain. This pathway is called the retinohypothalamic tract. It is worth sitting with the simplicity of this arrangement. The retinohypothalamic tract is not a branch of the visual system. It does not connect to the visual cortex or the thalamic relay stations that process sight. It runs straight from the retina to the clock, carrying one piece of information: light is present or it is not, with no elaboration and no context. The eyes are not only cameras. Buried inside them, alongside the apparatus of vision, is a light detector whose only job is to tell the brain what time of day it is. When the retinohypothalamic tract fires, it releases two chemical signals at the suprachiasmatic nucleus. One is glutamate, the brain's most common excitatory messenger. The other is a less common molecule, pituitary adenylate cyclase, activating polypeptide, whose name is rarely spoken in full, but whose effect on the SCN is quick and specific. Together, these signals trigger the rapid induction of two particular genes inside the nucleus's cells, period 1 and period 2. These genes are part of the molecular machinery of the clock itself, and their swift activation by incoming light is the mechanism by which the external light-dark cycle sets and maintains the master clock's phase, anchoring the internal rhythm to the rhythm of the rotating Earth. The details of how these genes work belong to a finer level of description. For now, what matters is the architecture of the connection, the slender wire running from the retina to the clock, the morning light activating it, the clock reading the signal and adjusting, the body's internal time shifting by a small degree back toward alignment with the sun. The suprachiasmatic nucleus has been shown, with unusual directness, to be not merely involved in circadian timing, but to carry the timing signal itself. The demonstration came from experiments in which the suprachiasmatic nucleus was surgically destroyed in rodents, both halves completely eliminated. Animals that lost their suprachiasmatic nucleus in this way did not simply lose a particular circadian rhythm, or weaken all their rhythms slightly, or become harder to entrain. They lost all circadian rhythmicity entirely. Running wheel activity, which in an intact nocturnal rodent is tightly concentrated in the dark phase and nearly absent in the light, dissolved into temporal chaos. The animals ran at any hour. They ate at any hour. Their core body temperature, which in normal animals follows a precise 24-hour wave, flattened into something close to a constant. Their hormone secretion, their sleep-wake cycles, the entire distributed temporal structure of their physiology, all of it gone, replaced by formless, arrhythmic activity with no temporal structure remaining. This much established that the suprachiasmatic nucleus was necessary for circadian rhythmicity. What came next was more specific. Fetal tissue from the suprachiasmatic nucleus of one strain of rodent, a strain whose natural circadian period was slightly longer than 24 hours, was transplanted into lesioned animals of a different strain, one with a slightly shorter natural period. The lesioned animals, already completely arrhythmic, began to recover rhythmicity after the transplant. Their locomotor activity gradually reorganized around a new, detectable period, and the period they showed was not their own original period, nor a compromise between the two strains. It was the period of the donor animal. The transplanted tissue had reinstated rhythmicity, and it had done so on its own schedule, establishing that the suprachiasmatic nucleus does not merely enable timing in a passive way, but carries the intrinsic period of the rhythm within itself, the way a tuning fork carries its pitch, regardless of where it is placed or what room it rings in. There are two distinct zones within the suprachiasmatic nucleus, and they have different chemical identities and different roles in the clock's operation. The outer zone, called the shell, is dense with neurons that produce a peptide called arginine vasopressin. The inner zone, called the core, contains neurons that produce a different peptide, vasoactive intestinal peptide. These two chemical populations do different things, and their difference matters to how the clock operates as a whole. The core neurons, those producing vasoactive intestinal peptide, are the ones most directly reached by the retinohypothalamic tract, the ones that receive the light signal from the retina and respond to it first. They are the entry point for the external world into the clock, but there is more to their function than receiving input. Vasoactive intestinal peptide released from these cells acts as an internal synchronizing signal within the suprachiasmatic nucleus itself, keeping the roughly 20,000 neurons of the whole structure, all their slightly different individual rhythms, running in coordination. Without it, the neurons drift apart. Each one keeps oscillating on its own period, as the experiments with isolated cells showed it would, but the network loses its collective coherence, and the coherent population rhythm that gives the suprachiasmatic nucleus its power as a master timekeeper, weakens and fragments. Vasoactive intestinal peptide is the internal conversation that makes a population of independent clocks into a single unified one. The shell neurons are different in their reach. Arginine vasopressin, the peptide they produce, acts not only within the suprachiasmatic nucleus, but beyond it, released in rhythmic pulses into the cerebrospinal fluid that bathes the brain, and into the small portal blood vessels that connect the hypothalamus to downstream structures. It is a broadcast signal, where the vasoactive intestinal peptide is an internal synchronizer. Arginine vasopressin is a messenger sent outward, telling the rest of the brain and through the blood, the rest of the body, what time the clock currently reads. This distinction between the internal conversation of the core and the outward broadcast of the shell is part of what makes the suprachiasmatic nucleus function as more than a clock for itself. It is a clock for everything downstream of it. The thalamus, which regulates arousal and attention, listens for the vasopressin signal. The limbic system, which governs emotion and motivation, listens. The circuits that regulate cortisol release and body temperature and immune activity listen. None of them needs to know the mechanics of the molecular oscillation to read the time. They only need to respond to the rhythmic chemical signal that the shell sends outward into the fluid that carries it. The cerebrospinal fluid moves slowly. It is produced in chambers deep inside the brain, circulates outward across the surface of the brain and spinal cord, and is reabsorbed into the bloodstream over the course of several hours, a slow and patient medium for slow and patient signals. Into this medium, the suprachiasmatic nucleus releases its arginine vasopressin in a rhythm that rises and falls across the day, higher in the morning, lower in the evening, following the same arc as the firing rate of the neurons that produce it. Any structure bathed in cerebrospinal fluid is bathed at the same time in this chemical record of the time. It is not a precise signal like an electrical impulse arriving at a synapse. It is more like a tide, slower, broader, carrying the time of day, not as a sharp fact, but as a sustained condition, a gradual shift in the chemical character of the fluid that surrounds every brain structure, from the deep hypothalamus to the spinal cord. There is something deeply old about this arrangement. Long before there were nervous systems capable of representing abstract time, living organisms kept circadian rhythms. Cyanobacteria, single-celled photosynthetic organisms among the oldest life forms on Earth, maintain 24-hour rhythms in gene expression that persist under constant conditions, driven by a protein-based molecular oscillator with no nucleus and no neurons at all. The timing of biological life to the rotation of the planet is not an invention of complex organisms. It is one of the most ancient features of living systems, older than multicellularity, older than animals, older than the separation of plant from animal. What the suprachiasmatic nucleus represents in this long view is not the origin of circadian timekeeping, but its centralization, the solution that complex bodies found to the problem of coordinating millions of cells and dozens of organs, each of which retains its own ancestral clock, all of them needing to run in something close to the same time. The body that contains a suprachiasmatic nucleus is, in this sense, a society of clocks. The liver has a clock. The heart has a clock. The lungs, the kidneys, the skin, the gut, each of them runs its own 24-hour program, governing the timing of thousands of internal processes, from cell division to enzyme activity to hormone sensitivity. For all of these peripheral clocks to remain coordinated with one another and with the external world, Something must tell them what time the master clock reads. The vasopressin that flows from the SCN's dorsomedial shell into the cerebrospinal fluid is one piece of that signal. Hormonal cascades triggered downstream by the SCN's outputs are others. The network of peripheral clocks and the mechanisms by which they are set and kept in alignment form a vast subject in their own right. What the suprachiasmatic nucleus contributes to that subject is its position at the center, the one structure whose rhythm, if removed, collapses the coordination of everything that depends on it. It is worth dwelling for a moment on the scale of what those 10,000 neurons per side are actually doing. 10,000 neurons is not many in the context of the brain. The visual cortex alone contains billions. Even small structures like the hippocampus, where memories form, contain millions of neurons. The suprachiasmatic nucleus is by this count a tiny thing, a rounding error in the total cell count of the human brain, a community small enough that it would be unremarkable in many other contexts. And yet it governs the timing of essentially every physiological variable the body maintains across the day. Cortisol, which peaks in the early morning and shapes alertness, attention, and immune readiness, follows an SCN-driven rhythm. Melatonin, which rises in the evening and signals the onset of biological night, follows an SCN-driven rhythm. Body temperature, which falls in the late evening and reaches its daily minimum a few hours before the habitual waking hour, follows an SCN-driven rhythm. Blood pressure, growth hormone release, immune cell deployment, the sensitivity of organs to insulin, all of it oscillates across the 24-hour day, and the signal that coordinates those oscillations originates in two paired clusters of cells occupying less space than the point of a fine needle. The neuron in the glass dish, the isolated SCN neuron firing on its 24-hour rhythm without any external reference, carries in some sense the whole of this. Every physiological rhythm downstream of the suprachiasmatic nucleus, every coordinated pulse of hormone and temperature and immune activity that makes up the temporal architecture of a living body, traces its timing back to the same molecular oscillation that keeps that single isolated cell ticking away in its dish. The complexity is immense. The organization is intricate beyond easy description, but at its heart, the timekeeping depends on the same thing the isolated neuron depends on, a molecular process that runs in a loop with a period of close to 1 day that keeps going because of what it is, not because of what the world outside tells it to do. The world outside can correct it. Sunlight arriving through the retinohypothalamic tract nudges the clock's phase each morning, pulling its rhythm toward alignment with the local light-dark cycle and compensating for the small imprecision that accumulates when any self-sustaining oscillator runs without external correction. This nudging is gentle and incremental. The clock moves by minutes per day, not hours, and the stability of the resulting rhythm, anchored both by the internal oscillation and by the daily light signal, is what makes circadian timing so robust under normal conditions and so persistent even when the external environment is disrupted. A person kept in constant light or constant darkness will maintain a circadian rhythm for weeks, maybe months. The clock does not need the external world to keep running. It only uses the external world to stay calibrated. What would it feel like to be a neuron in the suprachiasmatic nucleus at the peak of its daily activity? The question is, of course, not answerable in any direct way. Neurons do not feel in the sense that whole organisms feel. But something about the image is worth holding. At the height of the subjective day, these cells are firing rapidly, their electrical activity at its maximum, the chemical signals they release flowing outward into the brain's interior with the most force they will have all day. The VIP neurons of the core are in full conversation with their neighbors, their synchronizing signal at its strongest, the population rhythm of the whole nucleus, as coherent and precise as it will ever be. And the shell neurons are releasing arginine vasopressin at their daily peak, the pulse of it moving out into the cerebrospinal fluid with a quiet regularity that the body around it cannot sense directly, but responds to everywhere. Then, across the long afternoon and into the evening, the firing rate begins to fall, not abruptly, not in a switch on and then off. In a slow arc, a descent that mirrors the arc of the sun, the neurons quieting degree by degree as the subjective day gives way to the subjective night. The vasopressin signal weakens, the synchronizing VIP falls back. The nucleus, which at midday was its most vocal and its most precisely coordinated, settles towards something like rest, still oscillating, still keeping time, but more quietly, with less output, with fewer signals flowing outward to the structures that read its message. This is the state the suprachiasmatic nucleus occupies through the hours of sleep, not switched off, not dormant, but quiet. The molecular oscillation inside each cell continues to run. The feedback loops of the molecular clock advance toward the phase that will eventually reactivate the firing. The nucleus is, in some sense, keeping time through the night, in the way that a clock keeps time, whether or not anyone is looking at it, whether or not the body it governs is aware of anything at all. Sleep comes and deepens and cycles through its stages, and the brain does its nighttime work. And throughout all of it, the suprachiasmatic nucleus runs its rhythm in the dark, its 10,000 neurons per side, cycling through their quiet phase, counting out the hours until the slow rise of activity that will begin again in the early morning, the firing rate climbing, the vasopressin signal strengthening, the clock reaching its next daily peak, and the body, everywhere downstream, responding. There is something to linger in that image of the SCN at night. The rest of the brain is doing extraordinary things in sleep, consolidating memories, clearing metabolic waste, cycling through its architectural stages, the cortex moving through deep, slow rhythms, and then back up into the lighter, vivid dreaming states. The body lies still, the breath slow, the muscles released, and at the base of the brain, just above where the optic nerves cross in the dark interior of the skull, the master clock runs on in its own small, steady rhythm, undisturbed by sleep, undisturbed by dreaming, counting out its 24-hour measure in the dark and the quiet, with the same unhurried precision it has maintained through every hour of every day of the life of the body that surrounds it. 2 clusters of cells, each one no larger than a grain of rice, resting in the deep hypothalamus above the optic chiasm, silent in the sleeping skull. They carry no images of the world. They have no direct experience of the hours passing. What they have is a molecular rhythm that runs on its own terms, shaped by the light that enters the eye and corrected by it daily, but driven at its core by something internal, something that emerged from the earliest chemistry of living things, and that, in the form the suprachiasmatic nucleus gives it, now governs the temporal organization of the most complex body on the planet. The vasopressin released from the shell of the suprachiasmatic nucleus moves slowly through the cerebrospinal fluid in the dark. There is no urgency in this movement. The fluid is unhurried. It carries its chemical cargo through the chambers of the brain, past the deep thalamic structures, past the hypothalamic regions that regulate sleep and hunger and temperature, outward toward the surfaces of the brain and the fluid spaces of the spinal cord, and eventually into the circulation, where it rejoins the body's blood and disperses. By the time it has been carried this far, the pulse that produced it is already hours past. The clock that sent it is already in the process of releasing the next. And the body, receiving this slow chemical tide, reads from it the same information that a traveler reads from the position of the sun, not a precise fact, sharply stated, but a condition, a quality of the present moment, a sense of where in the cycle of the day things now stand. The time is late, the night is deep. The clock has said so in the only language it has, and the body has listened, as it always does, in the slow, unhurried way of things that have been listening to this signal since before they had a name. Deep in the body, in the small hours before dawn, the liver is working. No signal has been sent to tell it to begin. No conscious thought has commissioned the work, and the organ sits dense and dark behind the right side of the ribs, and inside its hundreds of millions of cells, enzymes are moving through their appointed rounds with the patience of something that has never needed to be told what time it is, because time is written into it. Glucose is beginning its slow climb. Detoxification runs at its nightly peak, processing the residue of the day before. Bile acids cycle through their synthesis, and all of it proceeds on a schedule set not by the brain overhead, not by the light outside the window, but by a clock inside each hepatic cell, ticking in the chemistry of the cell itself, precise and autonomous, and older than any nervous system that might oversee it. This is the liver at 3 in the morning. It knows what time it is. The body is not a single clock. That is the first thing to understand, and perhaps the most quietly astonishing. There is a master timekeeper, small and precise, resting at the base of the brain, where the optic nerves cross, and that structure does hold authority, does send its signals outward, does anchor the whole system to the rhythm of the world outside. But authority is not the same as sole possession. Every organ in the body carries its own clock, built from the same molecular gears, running on the same approximately 24-hour cycle, synchronized in health, but capable of drifting, capable of diverging, capable of running on a schedule that answers to something entirely different from the light coming through the eyes. The liver answers to food. That single fact, quiet and precise, opens into one of the deepest things the body does. Stretch across the broad territory of the liver's chemical life, and something becomes clear almost immediately. This organ runs on a timetable of extraordinary detail. Roughly 1 in every 10 to 15 genes active in liver tissue shows a daily oscillation in its expression, rising at 1 hour, falling at another, cycling through its rhythm as reliably as the sun moves through the sky. Genes governing how much glucose the liver releases into the blood. Genes controlling how fat is built and broken. Genes for the enzymes that clear drugs and toxins from the circulation. The cytochrome P450 family, each one with its own peak hour. The liver is not doing the same thing at midnight that it does at noon. It is running a different chemical program at every point in the day, and that program is coordinated internally by the clock inside its cells. What shifts this clock is not light. It is the arrival of food. When food comes in the normal hours, when meals land during the active part of the day, when the feeding schedule aligns with the rhythm the light-to-dark world has established, the liver clock and the brain's master clock run together, harmonized, locked in phase. The liver's peak detoxification hour lines up with the period the brain expects wakefulness. The morning rise in glucose production lines up with the approach of dawn. Everything moves in the same direction, at the same pace, like two hands of the same instrument. Shift the food, move the meals to the resting phase, to the deep hours of the night, when the body expects to be still and dark and fasting, and the liver clock begins to turn. Not overnight. Gradually, over the course of several days, like a compass needle being drawn toward a new pole, the hepatic clock reorients itself. It follows the calories. It pivots toward the new feeding window, moving its peaks and its troughs, its synthesis and its clearance, toward the hours when food is now arriving. And by the end of that slow reorientation, the liver clock and the brain's master clock are running on opposite schedules, 12 hours apart, each of them accurate by its own internal measure, both of them wrong in relation to the other. The liver, in other words, has inverted its day. Two clocks inside the same body, telling different times. The heart keeps its own clock too, and the stakes inside the heart are of a different kind entirely. Each cardiomyocyte, each individual muscle cell in the walls of the heart, contains the same molecular machinery, the same interlocking proteins that cycle through their rhythm across the span of a day. These cells regulate their own metabolism, the rate at which they consume and produce energy. They adjust the proteins that govern how strongly they contract. They modulate the ion channels that carry the electrical signals of each heartbeat. And they do all of this on a circadian schedule, so that the heart at 2 in the morning is at a cellular level, a measurably different organ from the heart at 2 in the afternoon. The clearest window into what this clock does is what happens when it is taken away. In mice bred to lack the core clock gene specifically in their cardiac cells, the heart clock silenced while every other clock in the body runs normally. The heart ages in a particular way. Over months and years, these animals develop a form of enlargement and dysfunction that grows worse with time, the chambers gradually failing the work they were built to do. The heart clock, in other words, is not decorative. It is structural. It is doing something essential to the long maintenance of the muscle. And in the living human heart, running normally, the circadian program is visible from the outside. The resting heart rate reaches its lowest point in the early morning dark, somewhere between 3 and 5, a long, slow interval between beats, the chambers filling and emptying at the pace of deep rest. Then, in the hours before waking, before any light enters the room, before any alarm has sounded, before the body has moved, the rate begins to rise. Blood pressure begins its morning climb. The vessels shift their tone. This is not the body responding to getting up. This is the circadian clock inside the heart and its attendant systems beginning the morning program before waking has occurred, anticipating the demands of the day with the kind of preparation that takes hours, not seconds. Even when a person lies still and quiet through a long night in a darkened room, their heart rate follows its arc, lower in the deep hours, rising before the biological dawn, because the clock is running underneath all of it, independent of behavior, independent of light, independent of will, below the heart, behind the stomach, wrapped in the coiling of the small intestine, there is an ecosystem. The gut holds more individual living organisms than the body holds cells. They shift and divide and produce their metabolic outputs across the day in populations that are not static, that rise and fall in relative numbers, that change what they are making and releasing, that carry their own rhythm of activity entrained to the habits of the body they live inside. The gut microbiome is circadian, not by choice or design. Those words mean nothing at this scale, but by the deep fact that its members are shaped by the timing of everything the host body provides them, the timing of meals, the hour at which the intestinal environment shifts its chemistry, the rhythmic changes in gut motility and secretion that the host's own clock drives through the day. The microbiome follows the host, and the host follows the light. So in health, when the host body's clocks run together, when light sets the brain's master clock, and meals arrive in the waking hours, and sleep comes in the dark, the microbiome runs with all of it, its population cycling on a stable 24-hour rhythm, its balance of species and metabolic output synchronized to the body's need. Disturb the host's clock, and the microbiome is disturbed too. Simulate the schedule of shift work, shifting the light exposure and the sleep timing and the meal timing in the abrupt, irregular way that shift rotation produces, and within days, the balance of species in the gut begins to shift. The ratio of major bacterial populations changes. The intestinal lining becomes more permeable, less precisely selective about what it passes into the body and what it keeps out. And this disturbed community, transplanted from a clock-disrupted animal into an animal that has never been disturbed at all, begins to produce metabolic effects in its new host, glucose dysregulation, fat accumulation, even though the new host has never lost a night of sleep. The microbiome, in this sense, carries the record of the disruption. It is a living record, transmissible, a community whose balance encodes the history of the timing of life in the body that built it. That is the depth of what synchrony means. In the gut, the kidneys run their own circadian program, quiet and largely invisible, but doing something fundamental to what sleep makes possible. Throughout the day, the kidneys filter the blood continuously, billions of small filtration units called nephrons processing the plasma that flows through them, adjusting what to reclaim and what to release, maintaining the chemistry of the body within narrow and necessary limits. Glomerular filtration rate. The speed of that filtering follows a circadian rhythm, higher in the active phase, lower during sleep. Renal plasma flow cycles across the day. The transport proteins that move specific molecules across the tubular walls are expressed on a schedule, their genes rising and falling in time with the clock inside the renal cells. Among these proteins are the aquaporin channels, the structures through which water moves across cell membranes in the kidney. In the pre-dawn hours, as the body lies still and the night deepens toward its end, these channels quietly reduce the volume of urine being produced. The kidney, under the instruction of its own clock and the signals it receives from the body's broader circadian system, concentrates what it needs to release and conserves what the sleeping body needs to keep. This is why a healthy sleeper does not need to wake to urinate 4 or 5 times a night. It is not merely that fluid intake has stopped. It is that the kidney has shifted its program, adjusted its outputs to the hour, set its aquaporin channels to a nighttime configuration that makes unbroken sleep physically possible. This is organ-level timekeeping in the service of rest. When the kidney's circadian rhythms are disturbed, in shift workers, in animal models where the clocks are disrupted, the fine regulation of blood pressure that the kidney participates in begins to drift out of its normal range. Renal pathology accelerates under chronic circadian disruption in ways that go beyond simple sleep loss. The kidney's clock is not separate from its health. They are continuous with each other, each one depending on the other running well. Reach now towards something the body rarely announces, something that has no single organ and no obvious location, but that may be the most consequential dimension of circadian timing in the living body. The individual clocks in the liver, the heart, the gut, the kidney, and dozens more in fat, in muscle, in the pancreas, in bone. These clocks are not synchronized by default. They are synchronized by the signals they receive, and those signals have to be consistent, and they have to arrive at the right times to keep all these independent oscillators running together. The light-dark cycle sets the brain's master clock. Feeding time sets the liver and the gut and the pancreas. Temperature, physical activity, Social contact. Each one carries some weight as a timing signal for one tissue or another. Under ordinary conditions, these signals reinforce each other. They arrive at hours that are consistent, that point in the same direction, that confirm to each organ clock that it is running in phase with the others. Under conflicting conditions, they diverge. When feeding time and light time and sleep time all align, when breakfast comes in the morning and the night is dark, and sleep comes with the dark, the body is one thing, running in one time. When these signals split, when a shift worker sleeps while the sun is up, and eats at 3 in the morning and sees bright light at midnight, each organ follows a different signal, and the clocks of the body begin to drift apart. This is internal desynchrony, not a metaphor. A measurable state in which the liver clock and the SCN run on different phases. The pancreatic clock answers to a different schedule from the cardiac clock, and the fat tissue and the skeletal muscle no longer peak at complementary times. The organs of the body lose their shared time. The effects come quickly. In healthy adults, carefully studied in controlled conditions, 3 days of simulated shift work, just 3 days of the kind of schedule reversal that millions of people live continuously, is enough to produce measurable changes in postprandial glucose, the rise of blood sugar after a meal. Blood pressure creeps upward, markers of systemic inflammation begin to rise, faint but detectable in the blood. 3 days, in people who started the experiment with no metabolic disorder, in people whose clocks were intact and well synchronized the week before. The body knows the difference between synchronized time and broken time, and it knows quickly. Return now to the liver, because the liver is the place where the consequences of desynchrony are most legible, and because the liver running 12 hours out of phase with the brain carries a particular image that is worth holding. Picture a building where every room has a clock, and every clock is set and maintained by a single reference clock at the center of the building, not through any wire or signal, but through the rhythm of when the building heats and cools, when people arrive and leave, when meals are served in the common room. For years, all the clocks in all the rooms run together, each set by the same shared conditions, each keeping the same time within minutes of the others. Then the schedule changes, for one room only. That room begins receiving its meals at midnight. The light comes on at different hours, and gradually following its own signals, the clock in that room drifts. Not because anything is broken, because it is following what it knows, the arrival of food, the change in light, the cues it is actually receiving. Within days, it reads a different hour from every other clock in the building. The liver, in a body living on the wrong schedule, is that room, and the whole building is trying to run on 2 times at once. Glucose metabolism works on one clock. Insulin secretion from the pancreas works on another. The proteins that carry fuel into muscle cells follow a third. These processes are built to interlock, each one set to peak when the others are ready to receive what it produces, each one set to fall when the cycle turns and the next phase begins. Slide them apart by hours, and the interlocking fails. Glucose arrives when the cells are not ready to take it in. Insulin rises when it is not needed, and flattens when it is. Fat metabolism and carbohydrate metabolism, which are meant to alternate on a circadian schedule, begin to overlap or gap in ways that favor accumulation over clearance. None of this is obvious from the outside. The body at the desk, the body on the night shift, looks like a body doing its work. The internal timing disorder makes no sound, causes no sudden event. It accumulates like the adenosine that builds in a brain that is not sleeping enough, slowly and without announcement, toward consequences that emerge over months and years rather than hours. The pancreas follows its own clock with a precision that shapes every meal. Insulin secretion is not merely a response to the glucose that food delivers. Its magnitude, its speed of response, its effectiveness, all of these vary across the 24-hour cycle according to the pancreatic clock. The same glucose load delivered to the same body at 8 in the morning and at 8 in the evening produces different insulin responses, different rates of glucose clearance, different downstream metabolic outcomes. Not because the food is different, but because the clock position is different. The pancreas is calibrated to the time of day, and so the body's handling of a meal is at least in part a function of when the meal arrives relative to the circadian phase. This is why it matters that food comes in the light phase, in the active hours when the pancreatic clock is set to its peak responsiveness, and the liver clock is in its phase of readiness, and the muscle clock has positioned its transport proteins to receive fuel. These organs were built to work together at these hours. The circadian system coordinates them, phase by phase, so that eating during the day means eating into a body prepared to handle the meal. Eating in the deep night means eating into a body that is, by every internal clock, in a different mode entirely. Fat tissue has its rhythm. Adipose cells, the cells that store and release fat, cycle through phases of lipid uptake and lipolysis on a circadian schedule. The genes governing these processes rise and fall across the day in patterns set by the clock inside each adipocyte, and entrained by the feeding and fasting signals the tissue receives from the broader metabolic landscape of the body. In the active phase, fat tissue is prepared to release stored fuel into the circulation when needed. In the resting phase, the default tilts toward storage, the cells prepared to accept rather than release. Skeletal muscle shows similar timing in its insulin sensitivity, its capacity for glucose uptake, its mitochondrial metabolism. The muscles of the morning are metabolically different from the muscles of the midnight hours, not because the muscle cells have changed their nature, but because their clocks have moved them through a different phase of their program. All of these rhythms were calibrated together under the selective pressure of millions of generations of bodies that ate in the light and fasted in the dark and moved during the day and rested at night. They are not arbitrary. They are the shape of a deep coordination that the body expects to be maintained, and that it registers with measurable consequences when it is not. Place the pancreas and the liver and the fat and the muscle side by side for a moment, not as organs but as clocks, each one ticking on its own, approximately 24-hour rhythm, each one set by the signals it receives: food, temperature, hormonal cues from the master clock and from each other. In health, these 4 clocks show a coordinated timing. They are not all at the same phase, because they are not meant to be, because different phases allow sequential handoff, each organ active when the others have prepared the way. The coordination is not synchrony in the sense of all striking at the same moment. It is synchrony in the sense of a well-run relay, each element arriving at its position at the right hour, each transition prepared by the one before it. This is the living body's most ordinary achievement. It runs invisibly, every day, in every person, and it is invisible precisely because it works. The coordination does not announce itself. It produces no sensation. There is no moment in an ordinary healthy day when a person feels their hepatic clock and their pancreatic clock running in phase. What they feel instead is energy in the morning, hunger at predictable hours, an afternoon settling, and an evening easing toward rest. All of that felt texture is the surface of this coordination. The harmony underneath is the organ clocks, each one keeping its time, all of them keeping the same time. And when the harmony breaks, What is felt first is often not a sense that the clocks are wrong, but simply that the day is harder, that the food does not sit right, that the energy is absent when it should be present, that the night does not bring the rest it should. The person rarely thinks, my liver clock and my master clock are 12 hours out of phase. They think, I am exhausted, and something is not right, and I cannot name it. The body's distributed timekeeping is so pervasive that disrupting it produces symptoms whose source is invisible. Small intestine, large intestine, stomach. The gut wall has its clock too, regulating the waves of muscular contraction called peristalsis that move food through the intestinal tract. Gut motility is not constant through the day. It is higher in the active phase, lower in the rest phase, on a circadian program that prepares the intestinal canal to receive and process meals at the hours they are expected to arrive. When food arrives at unexpected hours, the gut wall's timing is off, and what should be efficient transit through a prepared tract becomes a slower, less effective movement through a tract that is in its low motility phase. Every layer of the gut wall, the mucus that lines it, the immune cells embedded in it, the enteroendocrine cells that release hormones in response to nutrients, all of these carry circadian programs. All of them time their activities to the expected schedule. All of them benefit from food arriving when they expect it and suffer some degree of inefficiency when it does not. Bone remodeling follows a circadian rhythm, the cells that build bone and the cells that resorb it, peaking at different hours, maintaining the dynamic balance that keeps the skeleton strong and responsive. Immune cell populations cycle through the blood in daily waves. The composition of the immune system in the circulation shifting across the hours of a day in patterns that reflect when threats have historically been most common, when repair is most efficient, when the body is most likely to need one kind of response versus another. The timing of immune function is circadian. The timing of inflammation is circadian. The timing of wound repair and cellular maintenance is circadian. The body does not do everything at once. It cannot. Resources are finite, and the energy required for immune surveillance and the energy required for active movement and digestion and social engagement are not all simultaneously available in full. The circadian system solves this problem by assigning functions to hours, spreading the full agenda of a living body across the span of a day and night, each process given its peak when it is most needed and its trough when something else takes priority. This is what the distributed clock network is. In its deepest sense, it is not merely a system for keeping time. It is a system for allocating the body's resources across time, a schedule that has been tuned over immense spans of biological history to fit the rhythms of a planet that turns on its axis once every 24 hours and that divides its surface into light and dark with a reliability that the living world has come to depend upon entirely. The body expects the planet to keep its schedule. It has built its entire internal architecture around that expectation. The trillions of cells in the liver and the heart, and the gut and the kidney, each one running its molecular clock, each one keeping its piece of the body's time, they are all, in the end, synchronized to the same rotation of the earth. There is a particular kind of quiet that comes from understanding this, not the quiet of something resolved or finished, but the quiet of something very large becoming visible from a great distance. All this coordination, running without oversight, without any central command beyond the soft chemical signals that the master clock broadcasts, and the organs receive, and the cells interpret. Each organ clock running in its own tissue, setting its own transcription program, tuning its own enzymatic peaks, and yet in health, in rhythm, in the aligned body, the liver at its peak hour, the heart completing its nightly descent toward its pre-dawn minimum, the gut microbiome cycling through its daily population shift, the kidney's aquaporin channels set to their nighttime conservation mode. All of these separate clocks are doing one thing. They are keeping the same time, not because anything forces them to, because the signals that set them are themselves coordinated, each one part of a web of mutual reinforcement that the body maintains as long as the world outside provides its expected rhythms: light in the morning, dark at night, food in the waking hours, stillness in the sleeping hours, the temperature of the room and the pressure of the mattress and the chemistry of the blood, all of it whispering to each clock the same steady information about where the day stands. The moment any of these signals diverges, the moment light comes at midnight, or food arrives at 3, or the schedule rotates across the week, the web of mutual reinforcement begins to loosen at its edges. One clock follows one signal, another follows a different one. The phases begin to spread apart, and the seamless coordination that was invisible in health becomes visible, first as a vague disorder of energy and digestion and sleep, and eventually as something the measurements can find and quantify. 3 days of simulated shift work, glucose rising after meals in a way it did not rise before, blood pressure up by a few points, something faint and inflammatory in the blood, a trace of the body's response to its own internal disorder. The dysynchrony is already present, already measurable, before the person has fully noticed it. This is the nature of the body's distributed timekeeping. It works so completely when it works well that it disappears, and it begins to fail so gradually that the failure is long underway before anything dramatic announces it. The organs keep their time in silence. The liver in the deep night runs its detoxification cycle without sound. The heart's pre-dawn rate crosses its minimum without any sensation the sleeper would name. The aquaporin channels in the kidney close their nightly configuration in the dark of the organ, invisible and exact. All of this is the ordinary background of a living body, the invisible administration of a billion small functions assigned to their proper hours, the whole architecture of biological time running beneath every conscious moment of every day, patient and precise and old. Internal desynchrony is what happens when those organs lose their shared time, when the coordination that is health begins to fray at the hours when the signals diverge. When the liver clock and the master clock in the brain run like two orchestras in the same concert hall playing different scores, each one internally coherent, each one correct by its own measure, and between them a dissonance that the body registers in the chemistry of its blood. That dissonance is quiet. It makes no dramatic sound. It simply accumulates in the slow way that all these biological processes accumulate, in the patient bookkeeping of a body that expected one thing and received another, and that is still in the dark making its best adjustment. The kidney conserves water, the liver runs its program, the heart completes its nightly arc, and somewhere in the cerebrospinal fluid that bathes the brain, the slow chemical tide of the master clock moves in its own rhythm, washing outward toward structures too far to hear it directly, carrying what it carries, which is simply the time of day, the single most important piece of information the body has, and the one it has been building its entire internal world around since long before there were minds to observe it. Deep inside a nucleus, in a cell whose walls are too thin to see with any instrument but the finest, two proteins are moving through the slow chemical dark toward each other. They are not moving with direction. Nothing in chemistry moves with direction. They tumble and drift in the crowded fluid of the nucleus, brushing past RNA and histones and hundreds of other proteins whose business lies elsewhere, until by the physics of proximity and complementary shape they find each other. One fits against the other along their helical flanks, like two hands pressed together. For a long moment, measured in the time of molecules, which is to say microseconds that feel like nothing, they hold, and then, still joined, they settle toward a strand of DNA stretched out in the dark and find a six-letter sequence buried in a long, unbroken scroll. They land on it, they stay. What happens next governs the next 24 hours of that cell's life. This is happening right now, in the liver, in the heart, in the neurons of the brain, and the cells lining the stomach, and the fibroblasts deep in the skin, in nearly every cell of every tissue, in every body alive tonight. Two proteins, pressed together against a short sequence of genetic code, setting a clock that was already running before the first light of this day arrived. The proteins have names. One is called clock, the other is called BML1. They belong to a family of proteins that fold into corkscrew shapes, and where their helices touch, the bond they form is strong enough to hold against the constant molecular jostling of a living cell. The six-letter sequence they rest on is called an E-box. There are hundreds of E-boxes distributed across the genome, each one sitting at the entrance to a different gene, waiting for this particular pair of proteins to arrive and switch the gene on. When clock and BML1 land on one of these sequences, the gene downstream begins to be read, and what comes off that reading sets the timer for everything that follows. This is the positive limb, the push that starts the mechanism. It will not go on forever, across the next several hours, in a rhythm that is slow by human standards, and almost leisurely by the standards of most cell biology. The genes activated by CLUC and BML1 begin producing their own proteins. These are called PAR1, PR2, PR3, CRY1, and CRY2, and for a long time after their genes are switched on, they accumulate quietly in the cytoplasm just outside the nucleus. They build up slowly, the way a tide builds. A few at first, then more, then more. The cell has no sense of this. No process in the cell is watching the count, but the count matters. When enough PEA-RNCRY proteins have gathered, they begin to find each other and join. Dimers form, two proteins linked. These dimers are more stable than either protein alone, and they carry within their structure a signal that says, "Enter the nucleus." And so they do, drifting back through the nuclear membrane and into the dark interior where clock and BML1 are still sitting at their E-boxes, still driving the transcription of the very genes whose products are now returning to silence them. The P or cry dimer settles against the clock BML1 complex and without violence stops it. The positive limb is pressed shut. The genes that were producing P or and cry go quiet. No new P or cry is made. The proteins already in the nucleus continue their work for a while, but they too will be dismantled. Slowly, over hours, their concentration falls. The nucleus empties of them, and as it does, the grip on clock and BML1 loosens. The promoters open again. The cycle begins once more. The whole arc, activation, accumulation, inhibition, clearance, reactivation, takes approximately 24 hours. Not because anything counts to 24, because the speeds of transcription, protein folding, nuclear transport, and degradation in this particular system happen to sum to a day. It is not designed, it emerged. And it persists because organisms whose cells kept this rough interval were better tuned to a planet that rotates once every 24 hours, and over a long time those organisms left more descendants. The name for this cellular engine is the transcription-translation feedback loop, which describes its structure more precisely than its character. Its character is more like the turning of a wheel, whose motion is generated by the very friction that also slows it. The clock runs because it is wound, and it unwinds as it runs, and the unwinding itself is what rewinds it. Every revolution is complete. Every revolution begins the next. In mice bred with the BML1 gene deleted, not damaged, entirely removed, the wheel stops. There is no compensation, no backup system that runs when BML1 is gone. The SCN, the brain's master timekeeper, falls silent in its circadian oscillation. The liver clock stills, the heart clock stills. The animals eat and move and sleep, but without the rhythmic organization that BML1 drives, their behavior scatters across all hours of the day and night in roughly equal measure, with no strong preference for any time. They also develop a range of metabolic changes over their lives. Insulin handling becomes impaired, body weight drifts, inflammatory markers rise. Evidence that this molecular mechanism is not only about timing in the narrow sense, but about the orderly sequence of metabolic processes across the day. BML1 is, as far as the molecular biology can determine, the single component whose removal collapses the entire system. Everything else in the clock can be perturbed, and the system still runs after a fashion. BML1 cannot be removed and leave anything circadian behind. A single gene, a single protein, joining a partner in the dark of the nucleus, settling on a 6-letter sequence, driving 24 hours of ordered cellular life. But the speed of this wheel is not set only by transcription and translation. There is another layer of control, one that works not on the genes but on the proteins themselves, and it operates at a scale so fine that only a very particular instrument can detect it. Casein kinase 1. There are two closely related forms. Delta and epsilon is an enzyme whose job is to add phosphate groups to specific sites on PER proteins after those proteins have been made. A phosphate group is a cluster of 1 phosphorus and 4 oxygen atoms, and it is vanishingly small. It is invisible to any method that works by looking. It changes the shape of the PER protein, only slightly. But that slight change is enough to determine whether the protein will be stable for another few hours or be tagged immediately for destruction. When casein kinase 1 adds phosphate to the right site, a different enzyme recognizes the tag and marks the PER protein for the proteasome, the cell's protein disposal machinery, and the protein is broken apart in minutes. When phosphorylation falls on a different site, degradation slows. The protein survives, it accumulates, the cycle's timing changes. This is how the speed of the clock is set, not by transcription rates alone, by the survival time of PER proteins in the cytoplasm, which is governed by tiny chemical modifications added by casein kinase 1. The wheel turns at the rate it does because PER proteins last roughly as long as they do between synthesis and destruction, and that duration is controlled by enzymes that work 1 phosphate at a time, 1 protein at a time, in the fluid of a cell too small to see. The evidence for how precisely this matters is vivid. In a colony of golden hamsters, all running on a roughly 24-hour schedule, one animal was found to run its activity cycle on a 20-hour schedule instead. Every day, it compressed into 20 hours with the other animals spread across 24. Its wheel running, its sleep, its body temperature rhythm, all compressed together, all stable, all internally consistent, but shorter than any other hamster in the room. When the gene responsible was found, it turned out to be a single point mutation in casein kinase 1 epsilon, one amino acid, in one position in one kinase. The mutation made the kinase slightly more active. Slightly more active meant PER proteins were phosphorylated more quickly at their degradation sites. Degraded more quickly meant PER returned to low concentrations sooner. And PER returning to low concentration sooner meant the inhibitory phase ended earlier, the positive limb reopened earlier, and the whole cycle ran faster. Not a lot faster, 4 hours per cycle, but consistently faster for the whole life of the animal, because the mutation sat in every cell from birth. This mutation is called tau. The hamster carrying it is called the tau mutant. And the whole sequence from a single amino acid change to a 4-hour compression of the entire behavioral day is one of the clearest demonstrations that the speed of the living clock is determined by chemistry down at the molecular scale, where single modifications on individual protein molecules determine how long a step in the cycle takes. There is a second set of players in this mechanism, and they form a loop that wraps around the first one like a second gear mesh around the central wheel. Two proteins called Riva-Erbafa and Riva-Erbabet are among the genes switched on by Cluck and BML1. They are nuclear receptors, proteins that sit in the nucleus and regulate gene expression directly, and their target is BML1 itself. When Rever-Erba and Rever-Erba-beta are present in the nucleus in sufficient concentration, they land on a region of the BML1 gene called the ROR-E site and suppress it, drawing the gene towards silence. Less BML1 is made. Less BML1 means less clock BML1 dimer. Less clock BML1 means less activation of all the downstream genes, including Rever-Erba itself. As Rever-Erba levels fall, its suppression of BML1 eases. BML1 rises again. The positive limb restores. Competing with RAV-E-ARB at the same ROR sites are a family of proteins called RORs, RORpha, RORbeta, and RORg, which do the opposite. Where Rev-Reb suppresses BML1, Reb activates it. The two sets of proteins compete for the same binding site across the cycle of the day, their relative concentrations rising and falling in a pattern that reinforces the rhythm driven by the PER cry, negative limb. The clock is redundant in this way. Two loops interlocked, each one reinforcing the oscillation of the other. If either is disturbed, the other exerts a stabilizing pull. The system does not fail easily. Rev-Reb has a life beyond the clock, though its clock function and its metabolic function are not fully separable. In liver cells, Rev-Reb settles onto the promoters of genes involved in lipid production, in the making of glucose, and in inflammatory signaling, and it suppresses them at its daily peak. Its rhythmic rise and fall means that hepatic lipid synthesis, glucose output, and inflammatory gene expression all cycle in a Rev-Reb dependent rhythm across the day. The clock and the metabolism of the liver are not two systems that happen to be coordinated. They share components. The gear that turns the clock also moves the metabolic machinery. What times the cell also runs the cell. Reb rises to its highest concentration in the early evening, just as BML1 expression is being drawn toward its daily nadir. In a liver cell in the hours before midnight, Reb is sitting on hundreds of promoters simultaneously, drawing them towards silence in a wave that will ease only as Rev-Ret itself is cleared across the early morning hours, and then BML1 rises again, and clock returns to meet it, and the wheel completes another turn. There is something particular about the silence this creates, the nightly suppression of a clock's own most essential component that is worth resting in for a moment. The system keeps time partly by periodically stopping itself. It does not run continuously in the way a heart runs continuously. It oscillates by building to a threshold and then dismantling itself, building and dismantling, in a cycle that depends on the dismantling being as reliable as the building. The silence is not an absence of clockwork. The silence is the clockwork. For most of the history of chronobiology, the feedback loop was understood in the broad terms above: transcription and translation, inhibition and relief, roughly 24 hours per cycle. But biology never stays in broad terms for long. When researchers began measuring the proteins more carefully, tracking their concentrations across many cells and many cycles, they found something that complicated the clean picture. Different cells, given identical starting conditions, ran the clock at slightly different speeds. Not the same cell from day to day, which would suggest measurement error. Different cells from the same tissue, the same organism, with the same genome, running the same loop, but on periods that varied by an hour or more from cell to cell. The loop's output was not a single clean period. It was a distribution, a spread of periods around the mean of roughly 24 hours, and different cells landed at different points in that distribution. This spread is now understood to arise from the stochastic nature of gene expression, the fact that in a small nucleus, the production of any given protein is not a smooth, continuous stream, but a series of small bursts, some larger and some smaller, with random variation between them. BML1 is not expressed at a perfectly constant rate. It is expressed in pulses, each pulse producing a cluster of mRNA molecules, and the size and spacing of these pulses varies from cell to cell. The variation feeds into the timing of PER accumulation, which feeds into the timing of nuclear entry, which feeds into the period length. And so the period varies, not by much, but by enough to matter. In the body, this variation is managed by coupling. Cells in the same tissue signal to each other, coordinating their clocks into a coherent tissue-level rhythm that is more precise than any single cell could achieve alone. In the brain's master clock, this coupling is done by chemical signals passing between neurons. In peripheral tissues, it is done partly by signals arriving from the master clock and partly by local intercellular signaling, whose details are still being worked out. The clock is not purely intrinsic to each cell. It is partly a social structure, a network property, sustained by the communication between cells as much as by the loop inside each one. In humans, some of the clearest windows into the clock's molecular character come from individuals in whom a single component is altered, not deleted entirely as in the mouse experiments, but changed. One amino acid, one gene copy, too many or too few, in a way that shifts the period just enough to push the person's internal day out of alignment with the 24-hour social world. One group of people carry a mutation in PER2. The mutation falls in the region of PER2 that is phosphorylated by casein kinase 1, and its effect is to slow that phosphorylation, not at the degradation site, but at a site that controls when PER2 can enter the nucleus. When nuclear entry is slowed, PER2 accumulates in the cytoplasm longer before becoming available to inhibit clock BML1. The inhibitory phase arrives earlier in objective clock time because PER2 and its partners reach their threshold earlier in the day. The whole cycle shifts earlier. These people fall asleep early, reliably, often before 9 in the evening, and wake early, often before 4 in the morning. They cannot stay up later by choosing to stay up later. Their bodies are already in the falling phase of the cycle at the time most people are reaching their evening alertness peak. This condition is called familial advanced sleep phase syndrome. It runs in families because the PER2 mutation is inherited, following the pattern of a dominant trait. It is not a disease in the ordinary sense. The sleep itself is not disrupted. The total duration is normal. The architecture is intact. The clock simply runs on a different schedule, one set by the biochemistry of PER2 stability in the cells, and no amount of social pressure or light exposure has yet been found to durably shift it more than a small amount. The families who carry this trait live slightly offset from the rest of the social world. Their evenings end when most people's evenings begin. Their mornings start before the sky has lightened. They live inside the same 24-hour planet as everyone else, breathing the same air, looking at the same sky. But the clock they carry ticks on a schedule that is not quite aligned with the schedule of the world outside, and the offset is written into them at the molecular level in the phosphorylation site of a single protein. On the other side of the distribution, there are people whose clock runs long. Cry1 is one of the two cryptochrome proteins that form the inhibitory arm of the feedback loop. In the typical cell, Cry1 enters the nucleus with its PR partner and presses down on clock BMAL1 until it is cleared, at which point the positive limb reopens. The rate of TER-I1 clearance, how long it survives in the nucleus before being degraded, is a key determinant of how long the inhibitory phase lasts, and therefore of the total cycle length. If TER-I1 persists longer, the inhibitory phase extends, the positive limb remains closed longer, the cycle takes longer to complete. In people who carry a duplication of the CARE-I1 gene, not one copy, but an extra copy, producing more CARE-I1 than the typical cell, a variant form of CARE-I1 is made that binds more stably to clock BMAL1 and resists degradation more effectively. The inhibitory phase in these cells lasts longer. The clock's period stretches. In experimental studies, the period of skin fibroblasts taken from Cry1 duplication carriers and measured in cell culture runs at about 24 hours and 45 minutes rather than 24 hours, a difference that sounds small but accumulates. In the body, this extended period means the person's circadian clock completes each cycle slightly later than the day before. The sleep phase drifts, not by a huge amount, perhaps half an hour later per day under free-running conditions, but the drift is consistent, inexorable, and indifferent to the person's preferences or schedule. Left without anchor to fixed zeitgebers, the person's sleep onset moves progressively later each day, cycling through the full clock phase over weeks before returning to its starting point. With the anchor of work schedules and light exposure, the drift is partially constrained, but never fully. These people fight to fall asleep at ordinary hours. They lie awake in the small hours of the morning, their SCN running on a phase that says it is still late evening. They wake late, groggy, out of phase with the ambient world. This is called non-24-hour sleep-wake disorder. It is most common in people who are blind because light input, the primary anchor for circadian phase, is absent, but it also occurs in sighted people carrying the Cry1 duplication, and the molecular basis is now well enough understood that it can be traced to the half-life of a protein inside the nuclei of cells that have never seen light and have no way of knowing what time it is outside. The cell does not know what time it is outside. The cell only knows the time its own loop tells it, and if that loop runs long, the cell's time drifts from the world's time, and the person lives in the slow experience of that drift, always slightly out of phase, always slightly tired at the wrong hours, never fully at home in the clock that the rest of the world runs on. The loop does not know about mornings or evenings. It does not know about light. It knows only about the concentrations of the proteins it makes and destroys, and from those concentrations alone, it generates a period that is in most people close enough to 24 hours to be anchored by the day. A little fast, and external cues slow it down. A little slow, and external cues speed it up. But the cues can only work if the intrinsic period is close enough to grab. A clock running on 20 hours cannot be anchored to 24 by any reasonable environmental signal. A clock running on 28 hours will fight any attempt to hold it to 24. The loop sets the range within which the body can be entrained, and for most people, the range is sufficient. For some, by the accident of a single molecular change, it is not. What is present in a sleeping cell tonight, in a liver or a skin or a lung, is a loop that has been running since before that person was born, in a form essentially continuous with the loops running in the cells of every vertebrate alive and every vertebrate that has ever lived, back through the long cold time to organisms that had no ears or eyes or brain, only cells with this mechanism ticking in them, keeping a rough count of the planet's rotation. The proteins are different between a mouse and a person, not by much, but by enough that the period differs slightly, and the specific binding sites differ, and the post-translational modifications differ in detail, but the architecture is the same. Two proteins pressing together against a short DNA sequence, a wave of proteins building up and then dismantling the thing that made them, a period of roughly 1 day, arising from the kinetics of molecular biology rather than from any intention or design. The cell ticking tonight in the dark of the body does not know it is ticking. It has no sense of the rhythm it generates. It only produces proteins at the rates its chemistry allows, and degrades them at the rates its enzymes permit, and from those blind rates a period emerges. And from that period, the whole body's organization of time is built upward, layer by layer, until it reaches the level at which a person feels rested or tired, early or late, awake or drawn toward sleep. It is an old mechanism. It is older than sleep itself, older than brains, older by a vast distance than the concept of time. It is a feature of life's response to a rotating planet, written into chemistry somewhere in the long Precambrian dark, persisting because it was useful, and because what was useful was passed on. And it runs now, in the silence of a sleeping body, without any participation from the mind that is itself one of its products. In a single cell, Cry-1 is being cleared from the nucleus at this moment. Its grip on Cluck and BML1 is loosening as its concentration falls. Phosphate group by phosphate group, each one added by a kinase that has no knowledge of the day and no awareness of the night, until the threshold is crossed and the positive limb reopens and the cell's clock tips back toward morning. In another cell nearby, PER2 is crossing into the nucleus with its partner, pressed against the same clock, BMAL1 complex, beginning the slow inhibition that will run through the next several hours until PER2 in its turn is cleared and the cycle resumes. The two cells are not in perfect synchrony with each other. They never are, but they are close enough that the tissue as a whole oscillates together, their small individual differences averaging into a coherent rhythm that the whole organ keeps. And the organ's rhythm is close enough to the signal arriving from the brain's master clock that the two stay aligned across the hours, day after day, year after year, for as long as the organism lives. This is the machinery inside the life of any ordinary night. It is not felt. It does not announce itself. It runs in the darkness of the cell with the same patient indifference that the planet rotates, because the planet is what it learned from, in the only sense in which chemistry can be said to learn anything. The period is long, The mechanism is fine, and somewhere in a cell, right now, two proteins are finding each other in the dark and pressing together against a strand of DNA to begin another turn of the wheel, whose end will be its beginning, and whose beginning will be its end, and the day that follows will be organized, as all days are, by chemistry too small to see and too old to name, turning quietly inside the ordinary dark of a body at rest. In the first moments after sunrise, before the air has warmed, before the first sounds of the waking street have gathered into anything coherent, a single kind of light reaches the eye. It is not the brightest light of the day. It is not warm gold or the amber of afternoon. It is a pale, slightly cool illumination. The particular blue of the open sky before it has deepened into full morning, and within it there is a wavelength, near 480 nanometers, that carries a signal unlike any other the eye will receive all day. Most of the retina is built to receive images. Its cells are exquisitely shaped to detect edges and motion and color and the difference between a face and a shadow. They respond to change, to contrast, to the brief flash of a moving thing. But there is another population of cells in the retina, scattered among the more familiar ones, that are built for something entirely different. They do not respond to the shapes of things. They do not care about contrast or movement. What they respond to is simply the sustained presence of light, the long, slow accumulation of photons over minutes and hours, and the wavelength to which they are most sensitive is the pale blue of the open morning sky. These cells are called intrinsically photosensitive retinal ganglion cells, and they contain a photopigment called melanoposin. That name can settle to the back of awareness. What matters is what the cells do. When blue morning light falls on them and holds for long enough, they depolarize slowly, not in the sharp spike of an image-forming cell, but in a sustained rolling wave of electrical change. They are not telling the brain what the world looks like. They are telling the brain one single fact, that light is present, that it has been present for some time, and that the sustained fact of day has arrived. The axon of each of these cells does not take the path that visual signals take. It does not go to the visual cortex, where shapes are assembled and colors resolved. It travels instead along a dedicated tract, a narrow wire running from the back of the eye to a small paired structure at the base of the brain, a tract so specific in its destination, so indifferent to everything the visual system ordinarily processes, that it carries nothing the conscious mind would recognize as sight. What it carries is time, the raw, unadorned fact of morning. Deep in the brain, in the forward part of the hypothalamus, that signal arrives. The neurons there have been waiting in relative quiet through the night, their firing slow, their internal chemistry winding through the last hours of a cycle that began the previous morning. Now, with the arrival of the morning signal, those neurons begin to shift. Clock genes that track the pace of light respond. The chemical state of the cells changes. The cycle resets, or rather confirms itself. The internal rhythm, which has been running on its own close approximation of 24 hours, aligning once more with the world outside. The clock is not started by this signal. It was already running. The light does not wind the mechanism. It merely sets its hands. This daily alignment is the condition on which almost everything else in the body depends. The clock at the base of the brain needs to know when morning is, not approximately, not roughly, but with a precision that allows it to time the release of hormones, the drop and rise of body temperature, the slow architectural sequence of sleep, all of it measured against a fixed point in the day. Morning light provides that fixed point. It is the one signal the world offers that is reliable enough, stable enough, and rhythmic enough to serve as the anchor for a biological clock that needs to remain accurate over a lifetime. What the specialized retinal cells are doing as they receive the pale blue light of early morning is performing a task that has no analog in the rest of the sensory system. Hearing tells the brain what is making sound. Vision tells the brain what is in the world. But these cells are telling the brain something different: where the sun is in its arc, which side of the day this is, and how much longer the light will hold. They are not witnesses to the scene, they are witnesses to time. The light reaches them and holds. The signal runs its narrow, dedicated path, and somewhere in the brain's quiet, dark interior, a set of neurons responds to this announcement, and the clock of the body aligns with the clock of the world. This happens every morning. It has happened since the first morning, and it happens whether the person lying in bed is aware of it or not. The eye turned toward the window, the pale blue falling through the glass, and the body receiving the news of the day. By evening, that same clock is doing something different. The light has been withdrawing for hours, shifting from blue toward red and orange, and the sustained blue signal that has been suppressing a particular gland in the center of the brain is finally losing its hold. The gland is the pineal, a small structure shaped like a pine cone, which is where its name came from, though no one who named it could have guessed its function. Through the hours of daylight, the pineal gland has been held under a kind of chemical restraint. The light reaching the eye and traveling through a series of relays to the gland has been continuously suppressing the production of a molecule that belongs to darkness. As the light withdraws, the restraint loosens, not immediately and not all at once. The process is more like a slow tide going out. The molecule, whose name is melatonin, begins to be made in gradually increasing quantities from a precursor the body always has available. Serotonin, familiar from its many roles in the nervous system, is converted by a pair of enzymes into this new and different substance. The conversion is simple, chemically speaking, but its timing is everything. Melatonin is not a sleeping drug. It does not knock the body into unconsciousness or suppress the brain into rest. What it does is far more precise and, in its way, more interesting. It is a signal, a chemical message that travels from the pineal gland through the bloodstream to the brain and body, carrying one piece of information: that darkness has come. The body is full of tissues that are listening for exactly this message. Some of them use it to slow their activity. Others use it to shift the phase of their own internal clocks. The clock at the base of the brain receives it, and through receptors tuned specifically to melatonin, adjusts the timing of its own cycle. There is a moment in the early evening, roughly 2 hours before the hour at which a person habitually falls asleep, when melatonin reaches a threshold in the bloodstream. Researchers who study sleep call this moment the dim light melatonin onset, and it marks something real, the beginning of a biological transition that is larger than just the sensation of tiredness. Melatonin rising in the blood is the body's announcement that night has come, not to the brain's waking centers, which may still be fully engaged with screens and conversation and bright indoor light, but to the deeper, older systems that govern the timing of everything else. The heart begins to slow, almost imperceptibly. Body temperature has already begun its long nightly descent. The digestive system is easing toward its own quieter mode. Melatonin is not causing all of this, but it is one of the messengers through which the body coordinates these changes, a chemical tide that means night has come and the long preparation for sleep has begun. The rise of melatonin in the evening is not triggered by darkness exactly, it is triggered by the withdrawal of light. These are almost the same thing, But the distinction matters. If light were simply absent, melatonin would rise. But if light is present, if a person is sitting in a bright room at 9 or 10 in the evening, the signal that would allow melatonin to rise is still being blocked. The pineal gland remains under restraint. The evening transition delays. The body, receiving the message that day is still ongoing, holds its night chemistry at bay. Ordinary artificial light has some of this effect, but certain wavelengths are far more potent than others. The range that melanopsin cells respond to most strongly is the same range that dominates the screens of phones and computers and tablets, the blue end of the visible spectrum, near 480 nanometers, the same wavelength as the morning sky. A screen glowing in a dark room at 10 in the evening is, from the circadian system's point of view, very nearly indistinguishable from the open sky. The melanopsin cells receive the signal. They report that light is present and sustained. The pineal gland remains suppressed, and melatonin, which should by this hour be well into its rise, is held back. 2 hours of screen exposure in the late evening suppresses melatonin by roughly a fifth. The circadian phase shifts later. The body does not receive its chemical announcement of night on the schedule it expects. The transition that should begin at 9 is delayed toward 10 or 11, and sleep, when it finally comes, arrives later and shorter than the body's clock was prepared for. The waking the next morning comes at the same clock time. The alarm does not move, but the internal clock has been delayed, and the morning feels like something other than morning. The body is present in the day, but its clock is still behind it, running on a time that does not match the light coming through the window. All of this, the morning reset, the evening melatonin rise, the sensitivity to screen light, is the body's circadian system working exactly as it is built to work, in a world that no longer offers quite the right signals. For most of the time that biological clocks have existed, the light available at night was dim and warm and brief. Fire gives orange and red light, neither of which carries much signal for the melanopsin cells. Moonlight is dimmer than any indoor light. The human eye evolved in a world where, after sunset, the blue signal disappeared almost entirely, and melatonin was free to rise without interference. The modern evening reverses this. Blue light persists indoors long after it has vanished from the sky, and the body responds to what its sensors detect, rather than to what the sky is actually doing. Light is the strongest and most studied of the signals that anchor the circadian clock to the world, but it is not the only one. The body's internal timing can be influenced by a range of other signals, each one with the capacity to shift the clock, advance it, or delay it, depending on when in the circadian cycle the signal arrives. Food is among the most potent of these. Not food itself exactly, but the timing of eating. The clock in the brain responds primarily to light. The clocks in the liver and kidneys and pancreas respond primarily to the timing of caloric intake. When food arrives at the expected time, during the active waking phase of the day, it reinforces the alignment between the central clock and the peripheral ones. The body's organs receive both signals simultaneously. The brain's time announcement through hormones and nerve signals, and the liver's own detection of incoming nutrients. And these two pieces of information agree. The peripheral clocks and the central clock run together, pointing at the same hour. When food arrives at the wrong time, during the hours that were supposed to be rest, when the liver's clock was set to quiet its metabolic activity, the peripheral clocks begin to shift. Not immediately and not completely, but gradually. Over the course of days, the liver clock follows the meal schedule rather than the light schedule. In a mouse fed only during the hours it would normally sleep, the liver clock inverts almost completely over the course of a week. It has followed the nutrients rather than the light, leaving the liver running 12 hours out of phase with the brain, its enzymatic machinery active at the hours the rest of the body is trying to rest, and quiet during the hours when the body is awake and needs it most. This is a known problem in human biology. Shift workers who eat at night, travelers who cross time zones, and find themselves eating at local hours while their bodies insist it is still the early morning of their home. People who stay up late and take their largest meals after midnight. All of these are presenting their peripheral clocks with a feeding signal that conflicts with the light signal the brain is receiving. The result is not merely discomfort. Within days of this kind of conflicting signal, measurable changes appear in how the body handles glucose, how blood pressure behaves, and how the immune system responds. The meal timing signal is so powerful that when aligned correctly, when eating is restricted to the active waking phase of the day, it actively helps the body's clock stay synchronized. Time-restricted eating aligned to the waking hours reinforces the circadian signal rather than disrupting it. The liver clock and the brain clock receive consistent, agreeing information about where in the day they are, and the full network of peripheral clocks remains closer to the phase that keeps them working together. There are other signals as well. Regular social rhythms, consistent sleep and waking times, regular meal times, predictable patterns of activity, function as stabilizing forces for the circadian system, particularly for people whose internal clocks are prone to drifting. In certain psychiatric conditions, including bipolar disorder, circadian rhythms are inherently less stable, more easily shifted, more prone to losing their anchoring to the 24-hour day. Maintaining consistent social rhythms, eating at the same hour each morning, and sleeping at the same hour each night, building a reliable daily structure, provides the circadian system with a network of small consistent signals that together perform something like the function that morning light performs for the melanopsin cells. They tell the clock when it is, repeatedly and reliably, until the clock stabilizes around that information. None of these signals, light, food, social rhythms, are doing anything extraordinary. They are all simply providing the circadian system with the information it needs to stay aligned with a world that moves in a reliable 24-hour rhythm. The sun rises, the day has a structure, night comes. The clock that evolved inside the body evolved alongside this structure, and it expects the world to behave as it always has. What happens when the world no longer provides these signals in their expected form, when the light is wrong at night, or the meals come at the wrong hour, or the sleep-wake schedule shifts unpredictably from one week to the next, is that the clock begins to drift. There is a term for the mismatch between the body's internal time and the local social and environmental time, circadian misalignment. It is a condition most people have experienced at its most acute form, the state that follows a long journey across multiple time zones. A traveler who flies east across many meridians, from the western edge of one continent to the far side of another, arrives in a place where the local time is hours ahead of the time their body is keeping. The clock in their brain was set in the time zone they left. It is running on that time still, indifferent to the local hour, measuring the day from the last morning it was properly reset by local dawn light. The traveler's body insists it is, say, 3 in the morning. The city outside says 7 in the morning. The sun is fully up. The hotel breakfast is being served. And every clock in the body is on a schedule that has nothing to do with any of it. The discomfort of this state, the thick, unlocated tiredness, the inability to sleep at local night, the strange wakefulness at 2 and 3 in the morning, is familiar enough that it needs no description. What is worth sitting with for a moment is the mechanism behind it. The circadian clock at the base of the brain cannot simply be told that the time has changed and comply. It has no override mechanism. It has no ability to jump its phase forward by 6 hours because the local schedule requires it. The reset happens through the same mechanism it always happens through. Morning light reaching the melanopsin cells, traveling the dedicated pathway, arriving at the clock, and nudging its phase. A little each morning, a little more the next. Traveling east is harder than traveling west, and the reason is built into the clock's basic biology. The natural period of the human circadian clock, the length of its cycle in the absence of any external time signal, is for most people slightly longer than 24 hours. It runs a little slow. Left to its own devices, the clock would drift gradually later each day. This means the clock finds it easier to delay its phase than to advance it. Moving west, where the new local time is earlier in the clock's cycle than the old one, where the body needs to stay up a little longer to reach local bedtime, matches the direction the clock naturally wants to drift. Moving east requires the clock to advance, to run ahead of itself, to be pulled forward rather than allowed to slip back. The clock resists. It advances at roughly an hour to an hour and a half per day, which is close to the fastest it can manage. A traveler who has crossed 6 time zones eastward needs roughly 4 to 6 days before the body's clock has moved far enough to be genuinely aligned with local time. During those days, the internal and external schedules are running in parallel but offset from each other, like two pieces of music playing in the same room, but not quite together. Morning light helps, meal timing helps. Getting out into bright local morning light as soon as possible is the single most effective thing the traveling body can do to accelerate the clock's re-entrainment, because it is providing the strongest Zeitgeber, the German word for time giver, which researchers use for any environmental signal that entrains the clock at the right phase of the local cycle. The word Zeitgeber is worth holding for a moment because it captures something essential about the relationship between the body's clock and the world. The clock is not passive. It runs on its own. It has its own timing, close to 24 hours, generated by its own chemistry. But it is not sealed. It is designed to receive time-giving signals, to be adjusted by them, to stay coupled to the external world through them. Remove all the Zeitgebers. Put a person in a cave with no light cues, no meal schedule, no social contact, and the clock continues to run, but it drifts. It reveals its true period, which for most people is slightly longer than the day, and the sleep-wake cycle begins to slide slowly later each day, uncoupled from the sun. Provide the Zeitgebers, and the clock locks back on. Morning light is the strongest. Meal timing is the next most powerful, particularly for the peripheral clocks. Exercise at consistent times has a modest entraining effect. Social contact and the rhythmic structure of a regular day provide a softer but real signal. The circadian system is not rigid. It is a flexible mechanism that expects to be in dialogue with its environment and has evolved multiple channels through which that dialogue can occur. The traveler sitting in the early morning light on a hotel balcony, groggy and slightly displaced in time, is doing something that matters biologically. The blue morning sky is falling on their melanopsin cells. The signal is traveling its dedicated path. The clock at the base of the brain is receiving the local dawn and beginning to shift. 1 hour gained. Tomorrow morning, another. The adjustment is involuntary, unhurried, indifferent to what the traveler wants from it. It happens at its own pace, in the same way it has always happened, through the same cellular mechanism that has been resetting biological clocks since long before there were travelers to be displaced by it. There is a broader and quieter form of this displacement that affects a large portion of the working population without the drama of international travel. The human body has an intrinsic timing preference, an internal sense of whether early morning or late morning feels like morning, whether midnight or 10 in the evening feels like the natural end of the day. This preference has a genetic component, influenced in part by the same clock gene variants that set the period of the internal oscillator. Some people carry variants that shorten the clock's natural period slightly, and they tend toward earlier preferences, waking early, ready for sleep in the early evening. Others carry variants that lengthen the period, and they tend toward later preferences, slow to fully wake in the morning, more alert in the late evening and night. Neither of these is wrong. Both are expressions of the clock's natural variability. But the world is structured around a fixed schedule, and for people whose natural timing runs late, that schedule represents a daily misalignment. They rise at 7 because their job requires it, even though their clock is asking for 9. They go to bed at 11 on weeknights and then shift their sleep to midnight or later on weekends, partly recovering the sleep their schedule has been denying them, partly following the natural drift their clock always wanted. On Monday morning, rising for work feels like a 5 in the morning waking would feel to someone with an earlier clock. The mismatch between internal time and social time is called social jet lag, and it is estimated to affect somewhere between 40 and 70% of the working population to a meaningful degree. Social jet lag is not a dramatic condition. It does not produce the acute dislocation of crossing many time zones in a few hours, but it is chronic, and it is weekly, and its effects accumulate. And people who regularly experience a large gap between their internal clock time and their social schedule show associations with higher body weight, somewhat elevated rates of depression, and small but real reductions in cognitive performance. These are not consequences of sleep deprivation alone. Though sleep deprivation often accompanies the misalignment, they appear to be, at least in part, consequences of the misalignment itself, of running the body's many interlocked clocks on a schedule that does not match the one the environment is providing. The quietest and most common form of clock disruption is not jet lag or shift work or social jet lag. It is the gradual, nightly accumulation of blue light in the hours before sleep that has become simply part of the texture of modern evenings. The phone beside the bed, the screen in the dark room, the light that looks to the melanopsin cells like morning sky. The body does not know that the light is artificial. The melanopsin cells do not carry information about the source of the photons that fall on them. They respond to wavelength and intensity and duration, and in the late evening, in a room where a screen is glowing, those properties combine into a signal that the circadian system interprets the same way it interprets dawn. Melatonin remains suppressed. The chemical announcement of night is delayed. The body holds itself in a kind of extended metabolic day, waiting for the darkness that the screen is preventing. When the screen is finally turned off and the room goes dark, melatonin begins to rise, but it rises from a later point on the clock, and the phase of the whole system is pushed back. Sleep comes later. The alarm, unchanged, arrives at the same hour. The interval between the clock's sense of night and the imposed waking is a little shorter than the body prepared for. And then the next evening comes, and the screen is bright again in the dark room, and the cycle repeats. This is not a catastrophe. Most evenings with a screen do not produce significant harm. What they produce, accumulated over months and years, is a mild but persistent displacement, the body's clock running a little behind the day it wakes into each morning, the morning light arriving a little earlier than the clock expects it, the alignment between internal and external time never quite as clean as it would be if the evenings ended the way the sky ends them, with a long, quiet darkening and a warm dimness before sleep. The circadian system evolved in a world that offered consistent and reliable signals. Dawn came the same way every morning. Night fell the same way every evening. The clock that evolved alongside these signals is exquisitely sensitive to them, capable of tracking seasonal changes in day length over months, capable of adjusting its phase by fractions of an hour each day to stay coupled to the slowly changing sunrise. It is not fragile, but it is coupled, and what it is coupled to matters. In a room where morning light comes through good curtains, and the first light of the day reaches the eye early and blue and bright, the clock resets cleanly. In an evening where screens are dimmed and the light shifts toward warmer wavelengths as the hours pass, melatonin rises on schedule, and the body receives the news of night in time to prepare for sleep. These are not extreme measures. They are simply closer to what the clock evolved to receive. The traveler lies in the early morning light, not yet fully awake, the window pale with the blue of local dawn. They are 5 days into a new time zone, and the dislocation is mostly gone now. The clock has moved, one patient hour at a time, until it lines up again with the city outside. What they cannot feel is the alignment, the way the body's chemistry is now running on the same clock as the light, the way the cortisol has just begun its slow pre-dawn rise, and the melatonin is drawing down, and the systems that govern waking are beginning, in their own unhurried time, to open. The body does not tell the traveler that the clock has reset. There is no felt announcement, no moment of completion. There is only the morning, which feels like morning now, instead of the middle of the night, which is the whole of what the circadian system was working toward, the patient, unacknowledged return of inside and outside to the same time, the clock and the world agreeing again, quietly and without ceremony, on where in the day this moment is. The light continues to come through the window. The melanopsin cells receive it and hold their slow, sustained signal. Somewhere deeper in the brain, the clock confirms what it has already known since yesterday's dawn, that this is morning, that day has come, and that the long, careful architecture of the waking hours is ready to begin. The signal runs its quiet path. The clock, already running, settles into the day, and the light keeps coming, blue and unhurried, into the still room, carrying nothing but time. There is a weight that builds through the day, so slowly it is almost invisible until the evening arrives and the body finally names it. It has been accumulating since the first moment of waking, since the eyes opened, since the first breath drew in, since the first thought moved through the cortex and the neurons began their long day of firing. Not fatigue exactly, though fatigue is part of it. Something heavier than that and older. A chemical gravity, patient and cumulative, that does not announce itself but only deepens, hour by hour, until by the late evening it presses on the limbs and on the eyelids and on the quality of thought itself, and the body understands, at last, that it is time. That weight has a name and a substance. In the spaces between neurons, in the warm fluid of the basal forebrain, and deep within the tissue of the prefrontal cortex, a small molecule accumulates throughout every waking hour as a byproduct of the brain's ceaseless work. Neurons burn energy constantly, and as they burn it, they release this molecule into the interstitial space around them, the way a fire releases ash. It drifts and collects. Across the hours of a long day, it gathers in the narrow gaps where cells meet, settling onto receptors on their surfaces, slowing their signals, dimming their urgency. It is called adenosine, and it is the body's own record of how long the brain has been awake. Each hour adds to its concentration. The neurons keep firing, and the adenosine keeps rising. By mid-morning, it has reached a level the body can still manage, pressing lightly against the day's alertness, held back by the circadian tide that runs in the opposite direction. By mid-afternoon, a shallow trough opens in the circadian alerting signal, and the adenosine briefly dominates, a mild heaviness, a slowing of thought, a brief softness that passes. By evening, after a full day of waking, the adenosine concentration in the basal forebrain has climbed high enough that it cannot be ignored. It acts on two kinds of receptors, called A1 and A2A, and through them it suppresses the circuits that have been driving wakefulness all day. The wake-promoting neurons slow, the alerting signals weaken, the chemical tide that has been building since morning finally reaches the shore. Caffeine sits in this picture without changing it. The molecule that most people rely on to push against the afternoon's heaviness does not reduce adenosine or slow its accumulation. It only occupies the receptor, the same receptor the adenosine would bind, and holds the door closed against the signal. The adenosine continues to accumulate behind it, patient and indifferent to the blockade, so that when the caffeine eventually clears the receptor, the full weight of the morning and afternoon arrives at once. The tiredness was never postponed. It was only hidden, and the hiding has a cost. Somewhere in the lateral hypothalamus, a small cluster of neurons carries the task of holding the boundary between waking and sleep. The structure is barely the size of a small pea, and inside it somewhere between 50,000 and 80,000 cells produce two related neuropeptides, orexin A and orexin B, that project through long axons to nearly every corner of the brain, to the norepinephrine neurons of the brainstem, to the serotonin neurons of the raphe, to the histamine neurons in the tuberomammillary nucleus, and the dopamine pathways that run through the ventral midbrain. These are the wake-promoting circuits, and the orexin neurons hold them open like a hand holding a gate. Through the long hours of waking, the orexin neurons run at a steady output, driving the monoaminergic systems that keep the brain alert and stable. The signals they send upstream reinforce each other. The norepinephrine and serotonin in turn drive the orexin neurons, a mutually sustaining loop that makes wakefulness coherent and persistent rather than flickering. This mutual reinforcement is what makes it possible to stay awake through the afternoon slump, to think through fatigue, to hold attention when the adenosine is already high. The gate is held by the loop, but the orexin neurons are also connected to the circadian clock. They receive timing signals from the master clock in the anterior hypothalamus, and their output rises and falls on a roughly 24-hour schedule. During the active phase, through the morning and afternoon and early evening, they run near their daily peak. As evening deepens, the circadian signal to the orexin system begins its long, slow withdrawal. The gate does not slam shut. It closes the way evening closes, gradually the light going out of it, the tension leaving it degree by degree, until by the hour when sleep is expected, the gate has drifted nearly to rest. What happens to the brain when the orexin system fails entirely is one of the clearest windows onto what orexin actually does. In a condition called narcolepsy type 1, the cells that produce orexin are destroyed, not by age or injury, but by the immune system itself, which for reasons still being pieced together, turns against these particular neurons and eliminates most of them. With the gate gone, the boundary between waking and sleep becomes permeable in both directions. A person with narcolepsy does not simply fall asleep at inconvenient moments. They experience the complete dissolution of the border, sudden losses of muscle tone, triggered by strong emotion, a condition called cataplexy, vivid hallucinations at the threshold of sleep and waking, as if the dream state bleeds through the gap, and a sleep architecture that is fragmented and strange, with REM appearing almost immediately after sleep onset, rather than after the long gradual descent that healthy sleep requires. The gate between states, once lifted, does not easily return. There is a picture of sleep timing that unifies these two systems, the accumulating adenosine and the circadian clock, into a single account. The sleep scientist Alexander Borbély first proposed it in the 1980s, and its essential structure has held since. It describes two processes running simultaneously through every waking day, and it is their interaction that determines not only when sleep comes, but how deep it runs and how it is structured through the night. The first process is the homeostatic one. It is the adenosine curve, the slow, nearly exponential rise of sleep pressure across the waking day, accumulating continuously from the moment of waking, dissipating only during sleep itself, as enzymatic processes clear the adenosine from the interstitial fluid. This process carries no information about time. It only measures duration. It asks how long has the brain been awake, and the longer they answer, the higher the pressure. The second process is the circadian alerting signal, not a measure of how long one has been awake, but a measure of where in the 24-hour cycle the body currently stands. This signal is generated by the master clock and transmitted through the hypothalamus and into the arousal circuits, and it follows a daily arc that rises through the morning and holds elevated through much of the day before declining steeply in the late evening. Its peak is not at noon. It occurs counterintuitively in the late afternoon and early evening hours, the hours when most people feel most alert, because it has been climbing all day to counteract the rising adenosine tide. Without the circadian alerting signal holding the day open, the afternoon would be barely manageable. These two processes run in opposite directions, and for most of the day, they keep each other in balance. Homeostatic pressure rises, circadian alerting rises alongside it. Then, in the late evening, the circadian signal begins its steep decline, while the homeostatic pressure reaches its daily peak. For a window of several hours before this, approximately 2 to 3 hours before the habitual time of sleep, the circadian alerting signal is actually at its strongest relative to the adenosine pressure. This is the wake maintenance zone. Sleep during these hours is nearly impossible to initiate, even in a person who has been awake for many hours, because the circadian system is actively driving alertness, counteracting the homeostatic wait with its evening surge. The body is not ready. The clock says no. Then the circadian signal falls. It falls fast relative to how long it has been climbing. Within an hour or two, the declining alerting signal and the peaked homeostatic pressure cross each other, and at their crossing, the body experiences something unmistakable, not just tiredness, but a particular quality of tiredness that is nearly irresistible, that carries with it the sensation of the day completing, of the edges softening, of nothing in the world being worth staying awake for. This is the biological bedtime. DB came to be tame, in Wantadik Edidel. Both systems have been building toward it separately, from different directions, and they arrive at it together. The body does not lie down into nothing when it sleeps. It enters a structure, a precise and repeating architecture of states that cycles through the night in a particular sequence, governed partly by the homeostatic pressure that was so high at bedtime, and partly by the circadian clock that continues to run through the dark hours. The two processes do not switch off when sleep begins. They shift into a new mode of interaction, and their respective influences shape the night's interior landscape from one end to the other. In the first hour after sleep onset, the brain descends. The light, easily disrupted drowsiness of stage 1 gives way to the more stable sleep of stage 2, and then, within 30 or 40 minutes of lying down, to the deepest sleep of all. Slow wave sleep, sometimes called deep sleep or delta sleep, for the large, slow oscillations that roll across the cortex when an electrode is placed against the scalp. These waves, measured in the fractions of a hertz, sweep from the front of the brain toward the back in long, synchronized surges, and they represent something profound about what the sleeping brain is doing, a kind of global coordination, a slow consensus across billions of neurons that pauses the ceaseless processing of waking. Slow-wave sleep is the sleep of homeostatic debt. The higher the adenosine pressure at the time of sleep onset, the more intense and prolonged the slow-wave sleep in the early part of the night. After a night of full sleep deprivation, the first recovery sleep contains slow-wave activity of extraordinary depth and duration, as if the brain is paying back what it owes in the currency it was borrowing all day. During this slow-wave sleep, adenosine is being cleared. The enzymatic processes that were running against the tide all through waking are now running unopposed, and the concentration in the basal forebrain is falling hour by hour through the first long dark portion of the night. Something else may be happening during slow-wave sleep that is harder to observe, but no less fundamental. Along the channels of the glymphatic system, a network of fluid pathways that runs alongside the brain's blood vessels, expanding substantially during sleep. Cerebrospinal fluid moves in surges, coordinated with the slow oscillations, washing through the interstitial spaces of the brain tissue. The toxic proteins that accumulate during waking and are implicated in neurological disease are cleared most effectively during this slow-wave phase, in the steady flux of fluid that the sleeping brain's expanded glymphatic channels make possible. The brain cleans itself in sleep. The slow waves that look on an EEG like the idle murmur of a resting organ are also, at the tissue level, the rhythm of a deep and necessary maintenance. Roughly 90 minutes after sleep onset, the first slow-wave period completes its arc. The brain stirs, not to waking, but to a brief lighter stage, and then slides sideways into a different kind of sleep entirely. The large slow waves vanish from the EEG, replaced by a rapid low-amplitude pattern that resembles waking more than sleep. The body becomes deeply still. The voluntary muscles go slack in a kind of temporary paralysis. The eyes, behind closed lids, begin to move in rapid, irregular sweeps. The breath grows faster and less regular, and in the circuits of the brain, something close to waking ignites. The limbic and visual cortices lighting up, narrative and imagery and emotion running through the sleeping mind with the full sensory conviction of experience. This is REM sleep, and it is governed by different forces than the slow-wave sleep that preceded it. Where slow-wave sleep runs on homeostatic debt, REM sleep runs on the clock. The circadian system gates REM, holds it open or closed according to the phase of the 24-hour cycle, and this gating is one of the clearest demonstrations that the master clock does not simply time the onset of sleep, but continues to shape its interior throughout the night. The evidence for this comes from experiments in which people are kept awake for long stretches and then allowed to sleep at different circadian phases, or kept on unusual day lengths that separate the homeostatic pressure from the circadian signal. In these conditions, REM sleep consistently appears at the circadian time corresponding to the late phase of the night, the hours near and just before the habitual wake time, regardless of when sleep began or how long it has been running, even after 40 hours without sleep. After an extraordinary buildup of homeostatic pressure, the first long and vivid REM period still occurs at the circadian time of early morning. The clock keeps its appointment. Each 90-minute cycle through the night follows the same basic arc: a descent into some slow-wave sleep, then a period of REM, but the proportion of each shifts as the night progresses. The first cycle contains the deepest and longest slow-wave sleep and the shortest REM. As the homeostatic pressure falls through the first half of the night, the slow-wave sleep in each subsequent cycle becomes shallower and briefer. Meanwhile, as the circadian phase advances toward its REM gating window, the REM periods in the second half of the night grow longer and more vivid. By the fourth and fifth cycles, in the hour or two before waking, REM occupies most of the 90-minute window. The slow waves are nearly gone. The night ends in the state it began, with farthest from, in the vivid, emotionally colored, paralyzed but dreaming state that the clock has been saving for these hours. Through all of this, quietly and without any signal that reaches waking consciousness, the body's temperature is moving. Core body temperature follows a circadian rhythm with an amplitude of roughly 1 to 1.5 degrees Fahrenheit across the day. It reaches its daily peak in the late afternoon and early evening, in the hours when the circadian alerting signal is also at its strongest, and it begins its long descent in the hours before habitual bedtime, falling through the evening, through the first half of the night, reaching its lowest point somewhere in the pre-dawn dark, approximately 2 hours before the habitual waking time. This temperature rhythm is not a consequence of being asleep. It is generated by the circadian clock and would continue even if the body lay awake through the entire night. Its fall precedes sleep onset rather than following it, and the relationship runs both ways. Sleep onset most readily occurs on the falling edge of the temperature curve, in the window when the body's core is cooling, and the rate of this cooling is one of the better predictors of how quickly sleep will arrive. The cooling happens through the skin. The blood vessels in the hands and feet, the peripheral vasculature, dilate in the evening hours under the influence of the circadian clock, opening the capillary beds in the skin and allowing the blood flowing through them to release heat into the surrounding air. The hands and feet warm. They may become faintly flushed, and as they warm and release their heat, the core temperature falls, and with it comes a loosening, a heaviness, the particular quality of warmth around the edges, with a cooling center that is almost indistinguishable from the feeling of approaching sleep. In a warm bath taken in the early evening, this mechanism can be deliberately engaged. The bath heats the peripheral vessels, drives dilation, and accelerates the heat loss that follows. The core cools faster afterward. Sleep arrives more quickly. The body was going to do this anyway, on its own schedule. The bath only joins the process already in motion and moves with it. Lying still in the dark, the heat leaving through the fingers and the soles of the feet, the core temperature beginning its long pre-dawn fall. The body is preparing for something specific, not rest in the general sense. The precise biological architecture that requires and creates this particular temperature profile. Slow, deep, homeostatic sleep in the early hours, and then the long circadian gated REM of the late night, and then, at the temperature minimum, a brief cool stillness before the curve turns. That stillness is one of the quietest moments in the physiology of a human day. The core temperature has reached its floor. The circadian clock is in the phase that corresponds to the boundary between night and morning, though the sky outside may still be completely dark. The adenosine has been largely cleared through the hours of slow-wave sleep, the homeostatic debt substantially repaid. The last long REM period is running, the brain lit with imagery and narrative, the body still, the voluntary muscles held slack. The sleep is not deep in the way the early night sleep was deep. It is vivid and dreamed, and the body within it is almost motionless, resting its temperature at its lowest point, while the mind runs on in a different register entirely. Something about the relationship between slow-wave sleep and REM sleep mirrors something about the relationship between rest and meaning. The slow waves are restorative in the most literal and physiological sense, clearing waste, repaying metabolic debt, coordinating the maintenance of tissue and synapse, and the clearing of the glymphatic channels. They ask nothing of the sleeping mind, but that it be still. REM asks something different. It recruits the brain's full narrative and emotional machinery, runs it across the experiences and memories of the recent day, and produces something that waking could not produce, a processed, reorganized, emotionally integrated version of the day's experience. Whether this is the function of dreaming or only a correlate of it remains genuinely open. The experience of the dream and the function it serves, if it serves one, are not the same question, and only one of them has a clear answer. What is clear is that both are needed, and that the night, left to its own architecture, provides both in sequence and in proportion. The slow wave depth of the early hours descends from the high homeostatic pressure of a full day of waking, and the REM richness of the late hours rises from the circadian clock's long day of preparation, and together they make a whole thing. Neither one substitutes for the other. The first sleep of the night is not like the last. They are different states, doing different work, and the night contains both, only because the clock and the homeostatic drive happen to complement each other's timing when they are both running well. The oryx and neurons, in the hours of deep sleep, are quiet. Their long projections to the wake-promoting monoaminergic circuits carry no signal, and those circuits, the norepinephrine and serotonin and histamine systems that kept the day open, have dimmed to their nighttime minimum. The gate is closed. The consciousness that moved through the day's events has been set aside, not extinguished, and the brain's interior work proceeds in its absence. The adenosine clears. The glymphatic fluid moves. The slow oscillations coordinate the consolidation of the day's learning into long-term form. And somewhere in the anterior hypothalamus, the master clock continues its cycle, the molecular feedback loop still turning on its roughly 24-hour period, the vasopressin still pulsing from the dorsomedial shell into the cerebrospinal fluid, the time of day signal still broadcasting into every structure that receives it. The clock does not sleep. It runs through the night as it runs through the day, measuring with the same quiet precision, and what it measures as the night deepens is the approach of morning, not the morning that exists yet, the morning that the body is already preparing for, hours before the sky shows it. The cortisol rhythm, which carries the clock's morning signal to the adrenal cortex, has already begun its long nocturnal rise. The core temperature at its minimum now is close to the point where it will begin to turn. The oryx and neurons, their circadian input rising toward the morning phase, will begin to stir before the eyes open, re-engaging the wake-promoting circuits one by one, beginning the reversal of the night's long quieting. All of this, the slow waves and the REM cycles, the glymphatic clearing and the temperature descent, the orexin gate closing and beginning, hours later to open, all of it has been running on the interaction of two processes that have no awareness of each other and need none. The homeostatic pressure rises and falls by chemical accumulation and enzymatic clearance. The circadian signal turns on the molecular feedback loop that a small paired cluster of neurons in the hypothalamus runs continuously, indifferent to whether the body is awake or asleep, whether the eyes are open or closed, whether the day outside is cloudy or clear. The two processes do not communicate directly. They simply run, and their interaction in the nervous tissue of the hypothalamus and the brainstem and the basal forebrain produces, as an emergent consequence, the architecture of a night's sleep, its timing, its depth, its sequence, its proportion of slow waves to dreaming, its temperature profile, its duration, its end. Not one of these features is arbitrary. Each one falls out of the mathematics of the two processes, and the mathematics works because millions of years of refinement have settled the two processes into a relationship that serves the body's needs. Before anything had language for it, before there were words for homeostasis, or circadian rhythm, or adenosine, animals were sleeping on this architecture. Insects with simpler clocks and simpler nervous systems, fish that rested in the dark of the water, birds that tucked their heads against their wings and ran their own version of the slow wave and Argom cycle through the brief dark hours of the night. The fundamental structure predates the brain as we know it. It predates the SCN and the orexin system and the elaborate hypothalamic wiring that runs the human sleep-wake cycle. At its root, it is older than almost everything, the need to rest and consolidate and maintain, alternating with the need to be awake and active and present in the world. The clock and the debt turning together. The adenosine that presses now on the basal forebrain in these evening hours is part of a chemistry so ancient it is almost geological in its patience. It does not know that it is adenosine. It only accumulates and binds, signals the slowing of the circuits that have run all day, deepens the heaviness that has been building since morning, and when sleep comes, as it is coming now, it will not resist it. The enzymatic processes will begin their work. The molecules will be cleared, one by one, from the spaces between the neurons, and the pressure will fall, slowly and silently, through the long hours of the night. In the hands and feet, the blood is already moving toward the surface. The peripheral vessels are dilating in the way they dilate every night at this hour, releasing the body's heat through the skin in a slow and steady radiation. The core is cooling, the orexin gate is drawing nearly closed. The adenosine is high, and the circadian alerting signal is past its evening fall, and the two arcs that have been moving toward each other through the long day have very nearly crossed. There is a moment at their crossing, brief, exact, physiologically precise, when sleep becomes not just possible, but very nearly certain. This is not a thought the body has. It is a state the body enters, a threshold crossed by chemistry and timing, by the gradual quieting of the wake-promoting circuits, and the deepening of the homeostatic pressure, and the particular phase of the circadian clock. The moment is the same each night, adjusted each morning by the previous day's light, and the timing of the first meal, and the temperature of the room, and the 10,000 small zeitgebers of an ordinary human day. The clock resets itself, slightly, each dawn.